Thank you so much. I'm really delighted to be here today. So this is a video of a murine islet. It's expressing GCAMP6 in its beta cells, and it's been transplanted into the anterior chamber of a mouse eye. And so we can use this platform to functionally image islets longitudinally over weeks, months. And we've used this platform to start asking questions that interest us about coordinated or pan-islet beta cell connectivity and insulin secretion. And of course, that comes on the back of a huge body of literature that has connected calcium oscillatory activity at the islet level with insulin pulses in the systemic circulation. Of course, we know that the pulsatile nature of insulin secretion is really important for the normal physiological action of the hormone. And we know that pulsatility in terms of first phase insulin secretion and pulsatility per se is one of the hallmarks of the development of type 2 diabetes, so really important physiologically and pathophysiologically. And of course, we also now understand, given some of the talks we've heard this afternoon, about the really important paracrine signaling that occurs within the islet in terms of the control of insulin secretion. Okay, so we know at the single beta cell level the pathways that are associated with glucose sensing and insulin secretion, but arguably what is much less well understood is how all of the beta cells across an islet are coordinated, are working together in a connected way in order to subserve pulsatile insulin secretion. And those are the questions that we're interested in our lab. I just want to take you back a little bit and give you some context about some of the ways in which we think about beta cell connectivity in the islet. And this builds on some work from Maribor, so Andre Stozer, who 10 years or so ago now took slices of pancreata from rats, looked at them loaded with calcium dyes, and started to take out individual beta cell calcium time courses in response to the addition of glucose to those slices. And he undertook some correlation analysis, essentially Pearson R-based correlation analysis, looking at the calcium time courses between each beta cell in his cross-section and every other beta cell, and developed these now quite famous heat maps which describe pan-islet beta cell connectivity. And these guys were also the first people to note that connectivity in an islet follows small world network principles. In other words, there appear to be a small proportion of very highly connected beta cells. And of course, following on from that, we know of the work of David Hodson with Guy Rutter, who went on to suggest the functional relevance of these highly connected beta cells. They called them hub cells because the optogenetic silencing of hubs but not follower cells resulted in the subsequent diminution of pan-islet beta cell connectivity. Okay, so that raised a cynical eyebrow or two amongst the electrophysiologists, shall we say, and understandably. I mean, if you think about the electrical impedance of a system, it certainly can't be explained just through gap junctions alone, how one single beta cell, through electrical coupling, can augment or influence pan-islet beta cell connectivity. And so, rather thankfully, they gave us a let-off and suggested perhaps we need to think about intra-islet diffusible factors as a way of further delineating and understanding beta cell connectivity. Okay, so given that backdrop, we wanted to study the contribution of glucagon receptor signaling in beta cells on islet connectivity and pulsatile insulin secretion. So we developed a beta cell specific knockout of the glucagon receptor and looked at the effect of that on insulin secretion. And then as part 2 of the study, we went on to ask the question as to whether the pharmacological delivery of a glucagon receptor analog might in some way restore the lost beta cell connectivity that we see with type 2 diabetes. Okay, so I'm sort of now a little bit hesitant to give you this data because actually it's different to Jonathan's findings. But essentially we used Daniel Drucker's glucagon receptor flox mouse. We crossed it with our INS-CRE mouse, and so we have developed a mouse line with beta cell specific loss of the glucagon receptor, and we do indeed see loss of the glucagon receptor in islets, but no change in the expression of other incretin hormone receptors. The morphology of these islets of using IHCs appears to be completely normal, as do other markers of beta cell maturation, for example, the expression of disallowed genes. These mice breed very nicely. They have a normal body weight phenotype, but as I said, unlike what Jonathan has presented, we did find a subtle dysglycemic phenotype in these mice with selective deletion of the glucagon receptor in beta cells, as you can see here, particularly in the early stages of an IPGTT. Now, that suggested to us that there might be an insulin secretory deficit with this phenotype, and so we went on to look at that in more detail using a hyperglycemic clamp study, and again the glycemic controls in black and our knockouts in orange. So the control mice required a significantly higher infusion rate of glucose to maintain that clamped hyperglycemic, again confirming the phenotype of impaired insulin secretory profile. Okay, we looked, I haven't shown you the data, but we looked at the grouped 15-minute insulin levels in those IPGTTs. We didn't see absolute differences at 15 minutes and at 30 minutes in insulin levels when we took blood samples across the IPGTT. So we then thought to ourselves, well, maybe there's actually a slightly more subtle, nuanced effect of this phenotype in terms of insulin pulsatility or first phase insulin secretion immediately following the glucose bolus. So this here is a rapidly sampled, it's essentially a type of an FSIV-GTT. So here we give our knockout animals in orange, or our glycemic controls in black, a bolus of intravenous glucose, and then at the same time as transfusing back blood, because we had to so frequently sample, we sampled very, very frequently for insulin levels. And in essence, in our group analysis, we show a significantly altered burst mass pattern in terms of insulin pulsatility or pulses of insulin secretion in the first phase, following glucose administration, and some suggestion here that in the glucagon receptor knockout islets that we have this big burst of insulin release very early on, and then sort of a loss of those pulses which are retained in the littermate controls. Okay, so we now wanted to look at a little bit more detail what's actually happening in terms of at the islet level in these animals. So we then did another genetic cross. So we took our glucagon receptor in the beta cell knockout islets, and we crossed them with islets that are expressing GCAMP specifically in beta cells. So these are our control islets that are just expressing GCAMP in otherwise normal beta cells, and these are the islets that are expressing GCAMP in beta cells where the glucagon receptor has also selectively been removed. And perhaps you can see in this video here, this is at 12 millimoles glucose, you can just see these islets here, the knockout islets, look much less coordinated, and what we tend to see is this sort of rim of activity, but it doesn't quite end up as a pan-islet calcium wave, and perhaps that concept is better mapped out in these heat maps here, where every line across there represents a different beta cell region of interest, as in these perfusion studies the glucose level is increasing. We see this beautifully coordinated islet here, and in comparison, loss of the glucagon receptor in beta cells results in quite a profound loss of beta cell connectivity, at least in vitro. So far, I have suggested to you that loss of beta cell glucagon receptor results in a glucose intolerant phenotype, and that the glucagon receptor in beta cell plays an important role in synchronized calcium dynamics across the islet. Okay, so second part of the study, we wanted to ask the question, does administration pharmacologically of a glucagon analog restore some of the loss of beta cell connectivity that we see in obesity-induced dysglycemia? So here we take a group of wild-type mice. We transplanted normal islets, which are otherwise, normal islets expressing the G-camp fluorophore in their beta cells. We transplant those as reporter islets into their eyes, and then we took baseline readings of pan-islet connectivity here when the mice were still lean and metabolically healthy. We then repeated those imaging studies once the mice had become obese and dysglycemic. And then at that point, we divided those obese dysglycemic mice into 3 groups. So group 1 received 6 weeks of daily vehicle injection. Group 2 received daily injection of a long-acting glucagon analog. As you might expect, these animals will lose a certain amount of their body weight. So in order to try and tease out the body weight loss independent effects of the glucagon analog on islet function as measured in the anterior chamber of the eye, we had a weight matched to the glucagon-treated analog group as well. Okay, so here are our body weight results. So compared to vehicle-treated animals who actually continue to get fatter, we did indeed see about 10% body weight loss after 6 weeks in treatment with a chronic glucagon analog, and we managed to get a fairly good match with our weight-matched group. And interestingly, at the end of that 6-week study, both the vehicle-treated and the weight-matched group remained diabetic or dysglycemic, whereas we see a complete normalization of the glucose profile in the glucagon analog treated group. And then to try and understand to what extent some of that effect is related directly to islet function, we looked at our markers, our readout of pan-islet beta cell connectivity in those reporter islets in the eye, and indeed we see a diminution, as previously reported, of beta cell connectivity in response to high-fat diet and the development of type 2 diabetes in mice. We see that continue to happen in the weight-matched group, but the administration of the glucagon analog reversed some of that loss of pan-islet connectivity. So in conclusion, I hope I've persuaded you that intra-islet glucagon signaling is important for the physiological release of coordinated insulin from the islet. It's important for synchronized beta cell calcium oscillations, and in the setting of type 2 diabetes can restore synchronized calcium oscillations. And with that, I will very briefly thank all of my mentors and collaborators at Imperial College and beyond, and a particular warm thank you to Kinga Suba, who I didn't think was going to be here, but she has managed to come, and I'm sorry that you didn't get to give this talk because you really deserve to have done, having done so much of the work. Thank you very much.
Thank you, Victoria. Any questions? So I can then ask a question, because in your model where you surgically implant the islets in the eye, so those mice are alive when you're imaging, right? So what kind of sedatives are you using to keep the mice sedated? Because we know that both isoflurane but also other sedatives seem to affect islet function. And how much do you think that affects the connectivity you see in?
So we can still read differences in connectivity between low and high glucose. We use isoflurane, and we try to image them when they're fairly lightly sedated rather than deeply anesthetized, and as quickly as possible after the administration of glucose. But I agree with you, at the experimental level, although they're all getting isoflurane, it may well be having an effect on calcium activity. Nevertheless, I mean, we still get images like that, so they're clearly doing in vivo under isoflurane what they do in vitro in our perfusion experiments.
Thank you. Any other questions? Patrick?
Could you image many islets in the eye at the same time and really study this pulsatility? Ideally, you should do it in a streptozotocin-treated mouse.
Yeah, I've just asked for £500,000 for a microscope to do that, so I'll let you know. But I mean, the place to do it actually would be in the exteriorized pancreas, and to look at to try and look at many islets in the endogenous pancreas and tile that.
We tried to do that, but